them is different color, weird color. What color is that? Like pinkish or some beige, something like that, right? Anyway, red and blue is cardiovascular, but if it's pointing not to red and blue, you know that it's lymphatic system. Okay? So, but go over these structures so you have no problem identifying which ones which. Look for the thymus in your cat. You have to look for the thymus in the cat. You have to look for the spleen of the cat. You also have to identify the appendix of the cat and lymph nodes. You must know how a lymph node looks like. Okay? So here's our lab models. And by the way, if you are still maybe not confident with your A and P terms, now is a good time to go back to chapter 1. And please remember, one of the things that I remember was asked, like identify, right? Say this is, and you have choices, right? This is your lumbar lymph nodes, inguinal lymph nodes, popliteal lymph nodes, axillary lymph nodes. So you must know inguinal, very good. And you must also know that the direction of lymph is going up to the heart. Because the most common question is, where will this drain next? And I don't know why, and I'm telling you, students usually go from here down. They will circle, oh, it's going to go to my popliteal or something. It goes up, because it's supposed to pick up and return to the circulation. So it doesn't go away, but it goes up to join your circulation. That's why I said you must remember your A and B, your directions, your body regions. If you need a review, go look at that. I forgot what figure that is, but it's good refresher. Okay? And you will identify the lymph nodes in your cat. Look for some lymph nodes. They look like little beans, little kidney beans. When you identify, for example, the stomach or the heart, you're going to see some little kidney beans there. Those are lymph nodes. Lymph nodes are supposed to, in us, right? We can feel our lymph nodes sometimes. But when there's an infection, they're big and inflamed. It's very important to feel them. What is that clinical skill when you're feeling something? Palpation. It's very important to palpate them. Okay? Lymph nodes should be movable. When lymph nodes become hard and immovable, that's when you entertain malignancy. Usually, even when they're inflamed, they can still be moved. They're painful, but they can be moved. When you can't move them, that's when you have to request for biopsy. Okay? So you see, you have superficial and deep lymph nodes. The deep ones are located inside your body cavities. Look for some lymph nodes here. There's usually a lot of lymph nodes in your airways, just because, you know, you're inhaling always some impurities in there. Just be familiar with how they look like. So if I have it pinned, you know that I'm asking you to identify a lymph node. Okay? It's very sad how some people, right, still don't know their anatomy. So by now, I'm hoping that you know your major organs and where they are located. For your lymphatics, this is the small intestines of the cat. So now, when you open up the cat, you're free to open up, right? To look at your abdominopelvic organs right there. But don't pull away or sever them. This connective tissue is called the mesentery. And you're going to remember this term when you get to the digestive system. The mesentery is the membrane that makes sure those organs of your digestive system don't go anywhere. Even though you decide to swallow a whole chicken, I don't know, only snakes do that, I think. But no matter, right, they don't go anywhere. They're stuck there because they're held in place by the mesentery. So what I want you to do is you pull the mesentery and spread it like it's being done here. This is the cat. And you can see the red, right? This is your small intestines. The small intestines is the part of the digestive system where we absorb nutrients. And the red are your blood vessels, and that's where you absorb your glucose, your amino acids, all of the water-soluble substances. Identify the yellow one. Mm-hmm. The yellow one, right there, that's your lacteals. Lacteals, right? These are the lymphatic vessels there. And that's how the dietary fat makes it into the body. So what is contained in your small intestines are the digested nutrients. One of them will be the fatty acids. But fatty acids are insoluble in water. And your blood is mainly water. So the fatty acids will make it into the body through these lacteals. And while they're here, on their way to joining your cardiovascular system, they will be converted to lipoprotein. Are you familiar with HDL and LDL? We all know that. We say the good fat and the bad fat. So that's what's going on. The HDL and the LDL is the fat that has already been coated with protein so that it can join your blood. But if I pin this here and I ask you, okay, this is filled with what? There's a high concentration of what? Not glucose, not amino acids, but fatty acids. Triglycerides, you can write cholesterol, just fats. Of course, there's fluid in there. The content of the fluid, it should be called lymph. But the special lymph here is loaded with fat, so we call it chyle. Chyle is lymph, except it has a lot of fat. Everywhere else in the body, the lymphatic vessels will be filled with lymph, just plain lymph. This is also a type of lymphatic fluid, but it's just a ton of fat. So we change the name, we just say, okay, we'll call that chyle. Okay? So look for that in your cat. And then you will look for a lymph node. So like I said, we're going to ignore the slide on lymph nodes. I'm not going to use that. I may use a slide of the spleen. So there's your lymph node right here. This one, be sure you review the anatomy of a lymph node. I like to use this, right? And there it is. So you remember there's your afferent vessels and your efferent vessels. If you don't know, let's say, maybe I'll put a surface A is here, and this is surface B, right? Maybe I'll put A, B, C, whatever. And then I'll ask you, where is the point of entry of lymph? It's the one that has more vessels. That's the afferent. There's always more afferent than efferent, because that difference in number slows down the lymph flow, which is good because it gives the white blood cells time here to sort through everything that's entering the lymph nodes. How did the white blood cells make it there? Through this, right? This is your arteries, right? Arteries to arterioles. By the way, arterioles have no name. Just the big arteries, brachial artery, and then when you get to the smaller ones, they have no name. So this one will have no name. You just say, this is the arterial to a lymph node. And then you see when it makes it to the parenchyma of the lymph node, it will break into these capillaries. You see the capillaries have a red half and a blue half, right? So in the red half, there was net filtration. In the blue half, there was net reabsorption. My question is, do you think the white blood cells are being filtered out, or do they just move out on their own? Do you have to filter? They just move out on their own. That's right. Pressure does not affect their movement. So they have that amoeboid activity. They squeeze themselves out and make it here. What is being pushed out are all of the other nutrients, like your oxygen, your sugar. All of the cells that you see here need their own supply. But as far as the white blood cells, they're just able to move out. They have amoeboid movement, and they start populating your lymph nodes right here. That's how they make it here, okay? And so they're just waiting to sort through everything that passes through them, make sure that the lymph here has been filtered. So don't forget to include that in your studies, it's right there. There's a different set of white blood cells in the cortex compared to the white blood cells in the medulla, which I don't need for you to, it's just too much. Just white blood cells. But you need to know, if I ask you, okay, if the white blood cell here, there's a lot of B cells here, what do you think they will be doing? If there's a lot of cytotoxic T cells here, what are they trying to do? Or if there's a lot of eosinophils, what do you think is going on? So then you think, oh, if there's a lot of eosinophils, must be some worms in there or some allergy or something, okay? Okay. Oh, I'm not using this one. Next one. Okay, this one I always remind, because you are 200 level A&P. My blood pressure goes up. And I'm not kidding you, someone has identified this one to be the lungs. Right? So the key is the diaphragm right there. You know that above the diaphragm is the thoracic cavity. That's where your lungs are. That's where your heart is. It's impossible for this to be a lung or a heart or whatever. Students, identify that. In Bio 170, I'm more lenient because they're non-science majors. But in 200, you're going into the program. So it's very important for you to be able to identify these organs properly, okay? So there's your spleen. Be sure you identify that in all of our lab models. You only have one spleen, and look at the location. See the ribs right there? That's why if we do aggressive chest compression, or if there's a vehicular accident that fractured the ribs, this can very easily be lacerated because it's very vascular. You see all of those blood vessels, we just take it out, okay? But again, that's a major lymphatic organ that filters blood. Remember the difference between your spleen and your lymph nodes. The similarity is that they're both lymphatic organs. The difference is one filters lymph and the other one filters blood. You know which one is which, right? That's the difference. So because this is the only one really that will filter blood, if you have a patient, they have no spleen, you have to really educate them about being up to date with their immunization, etc., to avoid sepsis. So identify that in your cat, okay? And then look for lymph nodes. Be sure you can identify lymph nodes. Oh, I keep using this one. And then here's your spleen, and I also have this model exactly, this one right here. Be sure you know, identify this properly. This is one of those models that students sit down again and again and again. They don't know which one is which, right? Just to let you know, there's your liver, there's your gallbladder, right? This one, kidneys. This is adrenal. There's pancreas, and there's your spleen. Okay, so I don't know why they keep going back and they just keep turning it and turning it and turning it. They don't know which one is which tells me maybe they just ignored, then just went home, whatever. But be sure you include that. And then histology. So as far as histology is concerned, the spleen has both red pulp and white pulp. Because white blood cells have a nucleus, the white pulp is darker purple because of the purple staining nucleus of your white blood cells. And wherever you see an arteriole, that's where your white blood cells will be mostly concentrated because they get off, right? And they're pushed in the arterial end, so that's where they usually clump. And then when there's less white blood cells, also associated with venules, you're going to see more red pulp, red blood cells for red pulp. So be sure you train your eyes on how the spleen looks like. The lymph nodes actually look like the spleen, so I decided not to include that anymore to make it easier for you. Okay? So be sure you know which one is red and which one is white. The white is always associated with arterioles, and the red pulp is associated with veins. Oh, I keep using this one. Okay. And now, when you're looking at the organs in your cat, so what are you going to look for in the cat? Identify the thymus, identify the spleen, identify the lacteals, look for a lymph node. Also, a very important lymphatic organ is the appendix. Look for the appendix in your cat. In this lab model, I don't know, I hope you can see it, right? This is your small intestines. What you will do, you can see the diameter of the small. It's thin and small, right? That's why it's called small. Keep tracing, keep tracing, keep tracing until you get to the junction between the small and the large. At the junction, you're going to be shocked. You're going to look for the tiny, tiny, tiny appendix. You will put a star in your appendix to remember or to remind you that the appendix is not a digestive system organ. It has no digestion functions. It is purely lymphatic. And you should think about its strategic location. God put it there because you know your colon has lots of bacteria. And so the colon is the large intestines right here, and there's lots of bacteria here. But it's connected to the small intestines. The small intestines is where we absorb. So there's lots of blood vessels here. Now, because the small intestines is loaded with nutrients, right? And we know that the colon contains lots of bacteria. We all know that, right? There's lots of bacteria in the colon. The bacteria can be attracted to the nutrients up here. So they can just start creeping up. And when they creep up here, we know that there's blood vessels ready to welcome them, right? They can enter through the blood vessels here. This is very vascular because we absorb nutrients here. So to prevent that, you have the appendix. That's your checkpoint, Charlie. It's loaded with white blood cells. A tiny little thing can make it painful if you have an inflamed appendix, right? And even more dangerous if it ruptures. So look for that also in your cat. So you have cat dissection, you have that microscope, and then you have your lab models. And if you haven't taken blood pressure, please, or if you just need a refresher, please do that. I have an activity here if you want to do this, or if you just want to check blood pressure, you can do that. You got that? Those of you who haven't completed your blood ID, maybe you can use the rest of the time period to look for that, if there is still time. Okay? Be sure to wear your lab coats, your gloves, your goggles.